Okay, today I welcome Dr. Liam Crowley to the Enter Live webinar series to talk about unlocking invertebrate genomes and how Liam and a project that he's been working with or on are trying to sequence every species, but we're going to focus on invertebrates. That's been very clearly outlined on Twitter as well. So Liam, without further ado, I'm going to hand over to you and look forward to your talk.
Okay, thank you very much. Hello, everyone. It's an absolute pleasure to be with you today. And I am currently a postdoctoral researcher at the University of Oxford, and I'm working on this Darwin Tree of Life project. Some of you may have heard about it, some of you may not. That's absolutely fine. We're going to today just explore about the work that we are doing on this project. So to start off with, we're just going to go right back to basics and think about what is DNA and how does that relate to diversity, and then how are we sequencing DNA and the different techniques that we use to link DNA to biodiversity. Then we're going to have a little look at the actual Darwin Tree of Life project that we're working on, very exciting project that's going on at the moment. As well as specifically the work that I've been doing at White Woods next to Oxford, and sampling and trying to find all of the different species that we're sequencing. And then finally, we're just going to explore about why we're producing all of these reference genomes and what exciting stuff that they can tell us. So the Tree of Life project is a very exciting, very ambitious and collaborative program, which is trying to produce a full genome sequence for every species of eukaryote, so animal, plants, fungi, and protists, which occur in Britain and Ireland. Before we go into understand and look at that project, I thought I'd take us right back to the very basics. And when I mean right back, I mean right back. So about 4 billion years ago, there was a molecule, a complex polymer, so a big long chain molecule that began to copy itself and copy itself and copy itself and copy itself. And this was incorporated into the earliest life forms on the planet. And it was present in something which we call LUCA. LUCA stands for the last universal common ancestor. And from this single species, all life that we know of, and until we find some funky life on some distant planet in a galaxy far, far away, all life that does, and ever will exist is all based on this molecule, which began copying itself right back in the primordial soup of the Precambrian. And when Darwin was developing his theories of evolution by natural selection, he was actually remarkably accurate with determining how all life has branched out from this common ancestor. So you see his famous sketch on the left there, with showing the main branches of life. And I do love the annotation at the top, which just says, "I think." So even Darwin wasn't sure, but given the fact that he didn't know of the molecule that we're talking about at all, it's remarkably accurate. So of course that molecule is DNA, and that stands for deoxyribose nucleic acid, but that's not important. What is important is what it looks like and what it does. So it's essentially a really long chain with repeating structural sequences, and this structure inherently incorporates information. So it's a little bit analogous to how computer code is just a sequence of ones and zeros in binary code, which then tells the code and the computer how to work. The DNA is doing a similar thing, but inside of cells, and all cells, or almost all cells, have DNA inside of them, which is governing how the cells and therefore the organism grows, develops, and functions. And it does this based on the fundamental structure. So rather than having a binary code of 1 or 0 like a computer code, it has 4 possible options. And these are the nucleotide bases. So these are the little things that reach across the twisted helix, so like the rungs of the ladder. And we have adenine, thymine, guanine, and cytosine. Now, adenine always pairs with thymine, and guanine always pairs with cytosine. So we actually have two strands. But when we're talking about sequence, we're normally just talking about the one strand, the forward strand. But the order of those four, they make a code. And that code is divided into smaller sections called codons. And then that sequence is then read and interpreted by machinery inside of the cell, and then produces the function of that piece of DNA. Now, lots of DNA is what we call protein coding regions. So that develops into proteins by the process which we can kind of see a diagram of here, whereby the specific order of those bases, and you see on the left a little chart I made, that specific order then codes for a type of amino acid, and then the amino acids are linked together in a big long chain and folded in a complex 3D shape, and that's a protein. And proteins really make all of the processes in the cell work. But it's not just protein coding regions. Inside the DNA, there's also regions which turn on or upregulate other bits of DNA, and vice versa. There's regions which might inhibit other regions of DNA. And then you have these complicated cascades and systems where you can regulate DNA in all these different ways to make its function work. There's also loads of areas of the DNA which doesn't appear to do anything. In the past, this has been called junk DNA, but actually the more we look at it, the more we begin to discover that actually they are doing certain types of processes. There's also little bits of DNA which are almost kind of like parasitic and just kind of jump around genomes, inserting themselves and copying themselves. And in a sense, viruses are just escaped bits of DNA, which are doing the same kind of thing, but are able to escape from their original host, which is quite a crazy thought. So all of the DNA, which is contained within a cell or in an organism, altogether is called the genome. So that's all these genes, all these non-coding regions, and even stuff which is not in the nucleus. There's DNA which is held outside the nucleus, for example, in the mitochondria. And these genomes are enormous. So your genome, my genome, human genome is about 3 billion base pairs. So there's 3 billion of those letters in the sequence. And that encodes all of the instructions which to grow and function you as a organism. But this does vary. So some of the smallest genomes, certainly the smallest in the animals, are absolutely tiny, about 20 million base pairs. So orders of magnitude smaller than the largest. And the size of the genome doesn't necessarily correlate with how complex the organism is, or even how long ago it evolved. So there's all sorts of different processes going on which can affect genome lengths, such as huge chunks of the genome being duplicated. And actually the largest genome on the planets belong to the plants. So the largest of all is this Japanese flower, which is about 150 billion base pairs. But even in this country, mistletoe, which you may be familiar with, well, that genome is about 30 times larger than our own. So I like to think of the genome as a book. So a book is organized into chapters. So that's a bit like how a genome is organized into chromosomes. So these are discrete long molecules, and then they're all folded up and stored, because if they were unraveled, actually they'd be enormous, and they could go many, many miles. And then the chapters are then set in a series of paragraphs, and these could be a little bit like a gene. And in each individual word is analogous slightly to a codon, so that would be a sequence of 3 bases, which will then translate to the amino acid. And in each individual letter in the book, we have obviously the 26 letters of the alphabet, and in the genome we just have the 4 different bases. But they're not just static books. These are books which are changing all the time, because DNA is really subject to change. It's constantly getting damaged and fixed and repaired by the machinery of the cell. So for such an important molecule, it's actually remarkably dynamic. And this is a little bit like someone writing notes into the margin of the book, or potentially even ripping out a page and then inserting it somewhere else, or changing some of the letters. And sometimes this could change the meaning of the word. Sometimes it doesn't. And exactly the same in DNA. Sometimes these changes don't really do much, and sometimes they can be quite profound. So a population would be like every individual has its own book, which is broadly the same book as the other species, but its own copy might have slightly different notes in it. So when we're talking about genome sequencing, we're talking about decoding and translating this book. So imagine the book is in a different language and we're just translating it into a language that we can understand and study. So the process is by reading the order of all of those bases across the entire genome on each chromosome from one end called a telomere all the way up to the other telomere. So every single base in the right order. And this takes advantage of, there's lots of different sequencing techniques, but the original ones take advantage of physical properties of these different bases. So some of them use reflectance of the color of light, for example, and then Oxford nanopore, which is a more recent development, actually passes the strands of DNA through a really tiny, tiny pore. And then if we sequence all of the genome from an individual, that will produce a reference genome. So this is an example of a genome for this species, what it looks like. We can then even annotate that. So we can look at the genome and say, well, in this similar species, we know that this particular sequence encodes for this gene. So then we can start to mark on where we think the different genes and parts of the genome are. So assembling a genome is the second step. So the first step is actually reading out that sequence, but then you've just got a load of sequence and you still need to put it back together. So it's very similar to a jigsaw puzzle. And the original sequencing technology would have lots and lots of short fragments of DNA, which would then be read. And then we'd have to use very sophisticated software to arrange them back in order by looking at where they overlap and how certain we are that they were in that order. And a bit like how a jigsaw with a small number of large pieces is a lot easier than a jigsaw with a large number of smaller pieces, we can, with newer technologies called long read sequencing, we can start off with longer fragments of DNA, which means that we can be more certain that we're assembling them back in the right order, particularly because a lot of DNA is really, really repetitive. So it's difficult to know whether the bit you're looking at comes next in the sequence or actually has just come from another copy in a different cell. So it's a bit like doing a jigsaw, which is all the same color. And then once we've got all these overlaps together, we can then scaffold them together and reconstruct the chromosomes as they would be in situ. Now that's full genome sequencing, but that is a very different to DNA barcoding. Now, if you've heard of DNA sequences, chances are that was referring to DNA barcoding. So this is a particular type of DNA sequencing. And the idea is much like in a supermarket, each individual product has a unique barcode. There could be across the genome of all different species, a small region of it, which is unique to each different species. So it's a little bit like the Goldilocks porridge. It's got to be a sequence of DNA which is consistent enough across all of the species that you're looking at. So for example, the entire animal kingdom, that you can find it amongst all those fragments of DNA. You can recognize the motifs to find it, but it needs to differ enough that you can differentiate different species, but also not differ too much that different individuals from the same species look totally different. So fortunately, we have found some of these Goldilocks regions. So for animals, for example, we use a gene called cytochrome oxidase subunit 1. So this is part of a gene which encodes an enzyme actually in the mitochondria, so not in the nucleus. This is extranuclear DNA. And this enzyme is really important in respiration, which is a fundamental process by which organisms can generate energy. So it's really important. So if it changes too much, then it will be lethal. So it's really highly conserved across the entire animal kingdom. However, it has just the right amount of variation that different species usually have enough of a difference between them that we can distinguish them. However, that's not always the case. Just by chance, there are some species which might have either too much or too little variation to distinguish them, as well as there's some species which are still in the process of speciating, so we can't distinguish them as well. So when we sequence this, and you see down the bottom here, this is an actual CO1 gene from this bumblebee, Bombus hortorum. We get that barcode, and then we can compare it against a database which has examples of these barcodes from lots and lots of different species, and then hopefully it should cluster with lots of sequences. Now, these clusters, which are all identical or very, very similar, are called bins. That's why I've got a picture of a bin. It's not because I think it's a load of rubbish. And then usually a bin will correspond to a species concept. Now, it's not always the case, but a lot of the time that can be very useful to compare these bins and then determine what species we have. But it all relies on high quality reference material in the library. So that's DNA sequencing in a nutshell, but why? Why are we doing all this sequencing? Well, the first answer to that question is because we can, and actually it's getting a lot better at doing it and a lot cheaper. You can see here the cost to sequence a genome. For the first genomes which we did, which were hundreds of millions of dollars, we can now do it for significantly cheaper. And in fact, this graph is even now out of date, and we can do it even cheaper still. But just because we can do something is not always a good reason for why we should. So there's lots of different scientific motivations for sequencing a genome. And I like to think of them as two kind of categories. The first are called discovery science, by which we don't know what we don't know. We can find out all sorts of interesting things by looking at these genomes. And the second is what we could call enabling science, whereby genomes are so useful because they contain that entire history of not just that individual, but that species, as we can reenact evolution of that going backwards through time. So it's very quickly becoming an essential component of the 21st century biology toolkit. From things like developmental biology to physiology, even to behavior or even ecology, genomes can be really, really useful. And I've got some examples later as to why. But just to briefly expand on that now, here's some examples of some of the things that we can do with a genome. So we can look at how different species are related. We can look at how they've evolved. We can even look at how specific genes or groups of genes have evolved. We can also use it as a template to then resequence other individuals of the population to see how they are related or how they have evolved. And then finally, because the genomes are encoding all these processes to produce proteins, there's a huge potential for discovery of exciting new biomolecules, for example, which could be used in medicine, such as anti-cancer agents or even antimicrobial agents. You think about the diversity of life, all the fantastic solutions to particular problems that species have developed through evolutionary processes. It's very exciting to think the kind of discoveries that are out there waiting. So the Darwin Tree of Life is attempting to sequence loads of genomes. In fact, we want to sequence the genome of every single eukaryotic organism in Britain and Ireland. So every animal, every plant, every fungi, every protist, everything with a cell nucleus. And it's a very ambitious and collaborative project. It comes under this umbrella ambition project called the Earth Biogenome Project, where we want to sequence everything on the planet. But it's the first regional project which is trying to sequence the entire biota, or at least eukaryotic biota of a nation or geographic region. And as you can see here, here's some of the core partners involved with the project. So we have a range of institutions specializing in biodiversity and sequencing and data processing and analysis. And our kind of core aims on the project is to produce a chromosomally complete telomere to telomere, and in many cases annotated genome for every eukaryotic species in Britain and Ireland, which is about 70,000 plus, using PacBio long read sequencing, so very, very high quality genomes. As well, all of the data produced on this project will be completely publicly available. So anyone at any point can access that data. The sequencing technology we're using is long read on the PacBio Sequel 2, which will produce our sequences, and then we're using high-C long-range sequence mapping, so that will help with assembling those sequences into the correct order, so we can produce very accurate assemblies. Now, to produce all these assemblies, we need to get hold of the material. So this is the work that I've been more directly involved with. So this is trying to actually get hold of material from every single species. So the first thing that we need to do is really get an idea of how many different species there are, and what are we talking about. So we started off based on the UK species inventory, which is a checklist created at the Natural History Museum. And this has about 76,000 species concepts on it. However, and that's why I know there's an asterisk there, many of those species on that list were not seen for decades, or maybe they were an occasional migrant that got blown off course and was seen 50 years ago and hasn't been seen since, or incredibly difficult to find. So we know that the actual number is going to be somewhat less than that. So we have this definition of a species being within scope for us to try and sequence it is if it has a wild, outdoors, self-sustaining population. So it has to be breeding by itself outside. So that excludes things that might be introduced into hothouses and greenhouses, that kind of thing, or things which non-breeding individuals might occur very sporadically. What that actual number looks like is actually very difficult to get down to, and that's something we are working on at the moment. Now, eventually we want to sequence everything, but we have to start somewhere. So it was very important for us to identify our priorities. What do we want to sequence first? So initially, we'd like to have a very broad taxonomic representation. So we decided to sequence 1 or 2 species from every taxonomic family present in the UK. So we went and looked at every family and identified 2 species based on a hierarchical criteria. So the first of which, we wanted species which were important, maybe economically important, perhaps they are a pest or they are a ecosystem service provider, as well as iconic species, because things like stag beetle, for example, something that's really iconic, or they're interesting, scientifically interesting. So there could be researchers already looking at them. There could even be interesting stuff in their biology going on. Then after all those species, if there was none of those in that family, then we'd look to see, well, what can we actually get hold of? What's available for us to actually sample? There's no point picking a species if it's going to be really difficult to get, whereas there's a common relative which we can much more easily sample. Likewise, how representative is that species for that family? Have we just picked the weirdo, or have we picked something which is going to be interesting for us? I mean, sometimes actually the weird ones are interesting, so that would supersede it. And then the size, we actually, if everything else is equal, we would always go for the larger species, purely because that would yield more DNA from the extraction. So there's a greater probability of a successful sequence. And the genome characteristics as well. Again, if all things are equal, then we would go for one with a smaller genome, because there's a kind of an inverse relationship between the genome size and the amount of DNA that we can get out of and sequence. So if something's very, very small with a very, very large genome, it's actually really tricky for us to sequence. Likewise, there were other groups of taxa, not always taxonomically, sometimes just groups of related, so potentially ecological communities, but there were certain groups which we wanted to sequence, prioritize sequencing early on because they're interesting. So we call these deep dive taxa. So these are things like bumblebees and hoverflies as 2 examples. And these are, again, important, iconic or interesting groups. So once we know what occurs and we know what our priorities are, then we can go out and collect. And we've got 2 main models for our collections. The first of which is a kind of look at the list and go where it occurs and find it and collect it. So this is kind of like a diffused, targeted sampling. The other model is our site-based sampling. So this is based on our genomic observatory model. So we have a site, for example, Whiteton Woods, where I work in Oxford, is the first genomic observatory, where we want to sequence every single species that occurs in those woods, because we have all the systems and facilities in place to then process and those samples immediately, as well as the knowledge of the site, so we can begin to put those genomes into an ecological context. Once we've collected our specimen, we need to identify it, of course, to make sure it is what we think it is. Now, because we're doing long read technology, we need to have large fragments of DNA. And as soon as an organism dies, the DNA begins to break down into small segments within minutes. So we need to flash freeze everything from a live specimen to -80 to preserve it. So we need to identify it before it's frozen. So we started off with the low-hanging fruit and initially collected species where we could confidently identify them in the field. So a good example of this would be macromoths. We can do a lot of species of macromoths from field identification. Then, and we're doing this more and more, we have other groups of species where we can know roughly what it is on a field ID, but maybe there could be multiple species and we need to look at a certain character under the microscope. So then we use things like temporary knockdowns, either using cold or a blast of CO2, or sometimes even chemicals to temporarily incapacitate the individual. We can have a look at it under the microscope, see the features we need to see and identify it. And then the individual will revive and the DNA will be completely intact. So then we can flash freeze it to preserve the DNA for sequencing. Sometimes if that's not possible, but we know there's a key piece of morphology that could be used to identify it, for example, genitalia, as it's freezing, we could quickly dissect off that key bit of morphology and then look at that later whilst the rest of the specimen is preserved. Another method is to use conspecifics. So there are some instances where you have multiple individuals and we can be very confident that they are the same species. For example, we know they're siblings from the same brood, or maybe they're social species from a non-mixed nest. So then we can simply identify one individual using traditional techniques and then preserve and sequence another individual when we know they're the same. And then finally, we can also use DNA barcoding to identify species before the rest of the specimen then goes off for sequencing. So we would typically take a single leg. Now, every single specimen which we are sequencing, we are also barcoding, but this is not for a ID. This is to confirm the ID that we've already made and then also to assist with sample tracking. So once we've identified our specimen, we then need to preserve it. As I mentioned, because it's long read, we need to flash freeze it. So we need to get it from live to -80 as quickly as possible. So that should just completely isolate and freeze all of the various chemicals which could damage the DNA. So we do that using either liquid nitrogen in a dry shipper or dry ice, so solid CO2, as you can see in the photo here, which are about -80. So everything's cooled down and preserved. And then any specimen, large specimens, can be processed in the relevant way. Then we can barcode the leg that we removed, confirm the identification is what we said it is, and at the same time, it can be used for tracking. So the sequence which is produced at the end will have the barcode region in it. So we can look at that, match it against the sequence that we got at the start when we barcoded a leg, and check that there's been no mix-ups. At the same time, we can also confirm the taxonomy. Because we're doing so many different taxa, we do and will come across instances where the taxonomy is not resolved, or maybe there's different concepts. And everything is being based on the NCBI taxonomy, and every species concept assigned a unique taxon ID. So once we've produced all of these genomes, they are then published on the openly accessible European Nucleotide Archive. So anyone can go on and download and use these genomes. After it's been published there, we are also producing a short publication, a data note known as a genome note, which just explores and tells people how we sequence these genomes and some of the key metrics we get from analysis of the quality of the genomes, as well as having a short introduction introducing just the general biology of that species. So why are we doing all of these genomes, and what can they tell us? So the next part of the talk is all based on the work by my colleague, Dr. Peter Mulhare, who's another postdoc at Oxford, who's been looking at some of the genomes that we have produced on the Tree of Life project and asking some really interesting questions. So the first thing we can do with the reference genome, and this is one of the most common things that people jump to and ask about, say, oh, can we do this with the genome, is simply using it to resolve phylogeny. So simple molecular phylogenies typically would use a single gene region. For example, they could use a barcode region to look at the relationships between species. However, this doesn't always work. As I mentioned earlier, sometimes the barcodes don't differentiate between two species by chance, or perhaps they've diverged too recently. So we can use a full genome for a more in-depth process where we can actually look at multiple gene regions, or potentially, in some cases, even a whole genome alignments. So this is an example of one that Peter did for Lepidoptera, whereby we look at the genes which are shared by all of the species that we're including, and we look to see how they're related to each other. So as well as constructing the tree with how they're related, we can also see how these genes are evolving in these species. So the varying branch lengths, for example, will show the rate at which different groups are evolving. And by comparing those relative branch lengths, we can see actually some families, like the Lycanidae, the blue butterflies, are evolving much, much quicker than other families. Something else we can do with genomes is actually just study the genome structure. So we can look at things like the genome size. Remember, genomes are very variable in size, but even within particular clades, you get a lot of variation. You also get variation in how the genomes are organized. So things like the number of chromosomes that they split the genome into can vary quite a lot. And it's because it's changing all the time. Sometimes chromosomes can fuse, sometimes they can split. And surprisingly often, chunks of DNA can be taken, jumped from one chromosome onto a completely different chromosome. Something else which a reference genome is very useful for, in fact essential, is for population studies. So once you have this reference template, you can then use that as a basis for resequencing many individuals from a population. So a really good example of this is work which we are doing on the large blue butterfly. So the large blue butterfly went extinct in the UK, it's then been reintroduced from a Swedish population, and it's re-establishing and it's spreading. Now, as a species spreads, it's really important and useful for us to know about the genetic diversity within those populations to inform whether other management interventions need to be made, or just to kind of track and monitor that spread on a population genetic level. So to do that, we need to have a reference genome to know which loci, which part of the genome to look at. We can then very easily and cheaply take samples from lots of individuals non-lethally. So for example, we could do things like tarsal clips or wing clips, things which are not going to be detrimental to the individual. We can then sequence just that bit of genome that we've identified from the reference. So we don't need to worry about preserving big long fragments of DNA from a whole individual. So yeah, these kind of population studies are really important in conservation, and a reference genome is essential. And then the last kind of example of something we're going to look at for why and what we can do with the reference genome is by studying how species or genes have evolved. And this is arguably one of the most powerful and insightful things that we can do with the genome. So we've been doing quite a bit of this at Oxford with Peter's work. And one of the things we looked at was the Hox gene cluster. So the Hox gene cluster is a very important gene region which is involved in development. So in embryogenesis, the activation of these different genes tell the developing embryo its lateral positioning. So this part should be a head, this part should be a thorax, this part should be an abdomen. And there's a really famous, quite gruesome experiment where they've modified some of these Hox genes in fruit fly, Drosophila model. And then instead of growing antennae off its face, those cells don't know what part of the body in, so they end up turning into a leg. So you get these flies with a leg growing out of their face. So that's quite a dramatic demonstration of how important these homeobox genes are. And these are conserved across all animals, or most animals, including across insects. However, they are organized slightly differently in different groups. And there are some groups which have done some interesting stuff. And one of the examples of those is the Lepidoptera. So the very basal Lepidoptera, so the first ones to diverge and evolve, they have very similar Hox genes to non-Lepidopteran insects. However, most Lepidoptera have a lot of extra stuff going on. So if we plot all of these species across a tree, and then we put next to them a representation of all their Hox genes, we can see some quite dramatic reorganization of these genes. So many species have dramatic duplications where they keep copying and copying and copying certain these Hox genes. Now, Lepidoptera have done something quite interesting anyway, whereby they took this basal Zen second copy, and Zen duplicated and then duplicated again and again, so you've ended up with 4 copies, and these are called special homeobox genes. So these are unique to the Lepidoptera. And you see within these special homeobox genes, they then continue to duplicate in some lineages. And when we look at these special homeobox genes, we can see that they're very fast evolving, very quick rates of change within these special genes. So, and this quite often happens when you have these duplication events, because you have a gene, you then copy it. The original copy can just carry on doing the function that it's supposed to do, but that second copy is then free to change and evolve without having any detrimental effect, because you've already got a functioning copy going. So quite often these duplications lead to diversification. So that was the first way of looking at it, where we take the genome, we look for some interesting patterns, and then we try and work out what's going on. We can also do the reverse, and we can start off with the trait, and we find something interesting, and we can map it across the phylogeny, and then we can think, well, can we detect anything interesting going on with the genome, which is underpinning this trait? So we've done that here with Lepidoptera. We've done quite a lot of work on Lepidoptera because they're one of the first groups we really sequenced in depth, partly because of the ease of getting them. If we light trap, they come to us, and we can also identify many species with field IDs. So they were kind of one of the first groups, but it did mean that we had a huge number of Lepidoptera genomes, and we can start to ask these interesting questions. So once we've looked at the distribution of traits across this tree, we can then start to look at certain genes which we suspect might be involved with this. So for example, some moths or some Lepidoptera are diurnal, some are nocturnal, so we can look at opsin genes. And that's a work that's ongoing at the moment, where we're looking, and we found that actually within Lepidoptera, there are some really interesting duplications of a certain opsin gene related to production of light sensitive opsins receiving green wavelength light. Likewise, we're doing similar work looking at chemosensory related genes and their role in diet breadth. So this is it's a very big project, and I'm just one person involved. So there's an awful lot of people that I need to thank and acknowledge in all this work. So first of all, there's the Whiteham Genome Project team with our work on at Whiteham Woods, trying to get as many species as possible in the Genomic Observatory for sequencing. But also, this is just some of the people who have been involved with collecting and identifying samples from Whiteham Woods. There are more than this even, as well as the Whiteham Woods team for facilitating the research. Of course, got to thank the funders, the initial phase of the project funded by Wellcome. And this is just focused on actual sample acquisition. But as you've seen, it's a big, long process whereby there's many people involved downstream actually sequencing and assembling and producing these genomes, without which we wouldn't be able to ask any of these exciting questions. So yeah, I'll leave it there, and we can go on to questions.